Some examples of X-linked recessive alleles much more frequently expressed in hemizygous males are most forms of red-green color blindness, one form of the disease muscular dystrophy called Duchenne muscular dystrophy, and certain forms of the congenital disease which causes blood to fail to clot, called hemophilia, or love of bleeding. Hemophilia is notable as it spread throughout the European nobility in the 18th and 19th centuries. Again, note that these conditions have nothing to do with sex or reproduction without some imagination, I suppose. An interesting way to test for color blindness is by looking at the patterns of dots. If you are red-green colorblind, you probably don't see any numbers in these images, and you are almost certainly male, statistically speaking. Another interesting feature of X chromosomes is that all humans require only one functioning X chromosome to live. The normal case in many animals in XY sex determination is for females to have one functioning X chromosome and one that is dormant or inactive. Which one? It depends on the part of the body, but by and large, it is a random process which X chromosome gets inactivated. If a female is heterozygous for a gene that's encoded by the X chromosome, say for example coat color, different parts of her body will express different hair colors. We don't see this in human females, but we do see it in the animal that's featured in the background of the last few slides, the calico cat. The calico phenotype in cats is caused by X inactivation. The calico phenotype, in case you're not sure what that is like this nice puss down here. The X chromosome possesses a gene for coat color with different alleles, for example, orange and black fur. Developing inside mummy cat, the kitten had certain cell lines inactivate the X chromosome with the black allele and other cell lines inactivating the X chromosome with the orange fur coat allele. This leads to the mosaic phenotype we call calico. Because this phenotype is associated with this particular genetic phenomenon, male calico cats are extremely rare. It is said that the greatest showman himself, P.T. Barnum, offered a $50,000 bounty for anyone who could bring him a male calico cat, though I don't know if he knew about the biological principles behind the rarity, and I don't believe anyone ever claimed the money, but I don't really know. Here's something that I do know. This past summer, I adopted my kitty, Popoki, from the Lee County Humane Society. I also looked at some other cats on the website, including these kittens. I did not ask, though I am cultured enough to realize that these littermates were named after the characters on the Disney program Girl Meets World. Here are 2 males and 2 females. You can see that Riley and Maya both inherited the calico phenotype and that the black and orange pattern is random. They don't have the same pattern. Also note that Lucas and Farkle, the 2 males, each inherited a different X chromosome from their mother. Lucas is all black and Farkle is all orange. Biology in action. One more interesting thing about bar bodies is the range of different animals that may express mosaic phenotypes. In fact, the most bizarre one that I've ever seen thus far is a calico lobster. The Y chromosome is an odd bit of DNA in that it encodes very few genes. All other chromosomes encode many, many genes. As such, genes that are on the same physical chromosome tend to be inherited together, and we call them linked genes. After his initial success with the eye color mutants, Morgan produced many, many more mutant flies. Like Mendel, he performed a dihybrid cross experiment, and, like his first experiment, the results were surprising. Why? Because Mendel happened to be a little bit lucky in that the characters he studied, P shape and P color, were encoded by genes located on different chromosomes. Recall that his dihybrid cross led to the discovery of the law of independent assortment. But had he chosen different characters or an organism with fewer chromosomes, his results would have been even more difficult to interpret. But as we now know, genes located on the same chromosome are likely to be inherited together in this phenomenon called linkage. In his first dihybrid cross, Morgan crossed wild-type fruit flies with flies that had two mutations, black bodies instead of the wild-type gray, and misshapen wings he called vestigial instead of the normal flying wings that we saw in the wild type. The experiments started very similarly to Mendel's, flies with two dominant traits, gray body and normal wings, being crossed with flies expressing two recessive traits, black body and vestigial wings. As Mendel predicted, the progeny were 100% dominant phenotypes. But then Morgan took some of those F1 flies and performed what is known as a test cross. In a test cross, an organism expressing dominant phenotype is crossed with a recessive organism to test whether the dominant alleles are homozygous or heterozygous, though in this case, the hybrids were known to be heterozygous. The predicted results of the test cross are all four possible combinations of the two traits for the two characters. If the two genes for body color and wing shape were on different chromosomes, we would expect to see phenotypic ratios of 1 to 1 to 1 to 1, or 25% of each combination of body color and wing shape. If, however, the two genes were always located on the same chromosome and the gametes passed on by the parents were always inherited together, then the expectation would be that only the parental types would be present. What Morgan observed was neither of those things. A number somewhat in between equal distribution and all parental types, though with a strong bias towards the parental types. In other words, what he saw was mostly wild-type flies with gray bodies and normal wings, black flies with vestigial wings, and a few that were kind of mixed up between the two parent types. The results led him to ask, why? Why do gray bodies and normal wings develop together as much as black bodies and vestigial wings? And why, usually but not always? He reasoned that the genes did not assort independently, as Mendel predicted, and thus that the genes were inherited together because they were on the same chromosome. In other words, he had discovered genetic linkage. When the F1 offspring were crossed, the homologous chromosomes looked something like this. The mother would pass on the gray normal wing chromosome or the black vestigial chromosome. So most offspring would either have these chromosomes or these chromosomes. Most, but not all. How did these non-parental types arise? We know the answer, you and I, because Chapter 13 is behind us. But Morgan did not study Chapter 13, so he had to figure it out on his own. Back to Mendel for a second. He saw combinations of traits that were different from either parent. Hello, green round pea. Morgan called the flies that were just like the parents parental types, and the ones where the traits had rearranged recombinant types. If the genes encoding the two traits are on different chromosomes, the maximum expected difference would be 50% because of independent assortment. In Mendel's dihybrid test cross, there was a 1 to 1 to 1 to 1 ratio of phenotypes. 50% parental types, yellow round and green wrinkled, and 50% green round and yellow wrinkled, not consistent with what Morgan found in his flies. With the flies, Morgan found odd ratios because of linkage, but that linkage is not destiny. Some offspring were still recombinant. How? Hint, it happens in prophase 1 of meiosis, and it's crossing over. Crossing over can break the links between the parental genes and produce the different combinations, the recombination of those genes. How often does recombination occur? Again, the maximum expected value based on genes on different chromosomes is 50%. So less than that. We can calculate the recombination frequency by adding the total number of all recombinant flies, the ones not like the parents, and divide by the total number of all of the offspring, both the recombinants and the parental types. And this gives us a percentage which should turn out to be less than 50%, and it does. In this case, 17%. So that calculation, once again, is the number of recombinants divided by the total number of offspring. The top here is the actual data from Morgan's experiment, and the other expressions are generalized formulas to calculate the recombination frequency. I will not ask you to make calculations like this on the exam for this class, but your future genetics professor most certainly will. What is important for this class? Knowing that recombination frequency can be calculated, and it is less than 50% because of linkage. And one more thing that I'll get into in a moment. Recombinant chromosomes produce allele combinations that didn't exist in the parents, which is interesting beyond just what body color or wing shape you prefer in your particular pet fruit fly. Recombination makes never-before-seen chromosomes completely novel. Remember back in chapter 13 when I showed you some calculations of how unique you are, and I said we don't even take crossing over into consideration? Well, now we are taking that recombination into consideration, and it dramatically increases the variability in populations. Now, for that third thing you need to remember about recombination frequencies. So, Morgan demonstrated that genes are found on chromosomes, and then he showed that two genes can be linked on the same chromosome. Introducing Alfred Sturtevant, who was one of T.H. Morgan's graduate students. Sturtevant reasoned that if you can calculate recombination frequencies between two genes, you can figure out the recombination frequencies between 3 genes, 4 genes, or all genes. He was able to build a genetic map of all of the chromosomes with many, many, many mutant flies. Reasoning that the further apart two genes are, the higher the recombination frequency until it approaches 50%, like it would be if the genes were on 2 different chromosomes. What Sturtevant produced was a linkage map based on calculating hundreds of recombination frequencies. Now, these map units that he came up with are relative. We know that chromosomes are made of DNA, but the recombination frequencies are percentages that don't tell us how many nucleotides are between genes. It's kind of like if I were to ask you how far away Atlanta is from Auburn, and you might say it's 2 hours. That's a relative distance based on driving time and dependent on traffic and how fast you like to drive. An absolute measure would be to say that Atlanta city limits is 100 miles away from Auburn. That number is not going to change. So Sturtevant's method worked kind of like this. He produced many different mutant flies and performed recombination experiments. While the previous dihybrid cross showed that the black and vestigial wing genes had a 17% recombination frequency, a third mutant, a different eye color mutant he called cinnabar, had a 9% recombination frequency in crosses with the black body mutants, and about 9.5% recombination frequency when crossed with vestigial wing mutants. Thus, he was able to piece together the relative order of the 3 genes on the chromosome. More and more mutants were found and more recombination frequencies calculated. Today, we've been able to build upon the maps produced by Sturtevant and Morgan, and now we have a complete genomic sequence of the fruit fly and many, many other organisms as well, including humans.